Hi, I'm Elizabeth.
And I'm Sarah.
And we're your UTAs that will be talking to you about enzymes today. So here in this first graph, we're discussing how a reaction rate changes as you increase the concentration of an enzyme. As you increase the concentration of an enzyme, the reaction will begin to increase until a certain point at which it will level off. The leveling off occurs because the enzyme has used up all the substrate present in the cell, and the reaction cannot possibly speed up anymore until you add more substrate.
Okay. And also we can talk about how the concentration of reactant affects the rate of the reaction. So as before, it increases, and at a certain point it levels off. And the reason that it levels off is because the amount of enzymes are used up. So all of the enzymes are saturated at this point, and then increasing the concentration of reactant will not help. They're all saturated.
There are different molecules in the cell besides the original substrate that combine to the enzyme. This first type of inhibition is called competitive inhibition. So a molecule other than the substrate will bind to the active site of the enzyme. This prevents the intended substrate from binding to this active site because it is unable to bind to the site that's already occupied by the inhibitor. The way a cell can help address this issue is by increasing the concentration of the substrate. By increasing the concentration of the substrate, you increase the chance that the desired substrate will bind to this active site.
There's another type of inhibition called non-competitive inhibition, and here these inhibitors bind to somewhere other than the active site. But this still has an effect on the enzyme because it changes the shape of this active site. So the normal substrates will not be able to fit. So they too will not attach to the enzyme and the reaction will not occur. But instead for this type, increasing the concentration of substrate will not help because no matter how much substrate you have, you can't change the fact that it doesn't fit in the changed active site.
Enzymes function at different environments throughout the cell. So for example, one factor that can determine the efficiency of an enzyme is temperature. As you increase the temperature, your reaction will begin to occur faster because as the molecules move around quicker in the cell, the chance of them interacting and bonding to that enzyme increases. However, if you increase this temperature too much, your enzymes will begin to denature because you'll be breaking those ionic hydrogen and van der Waals reactions that hold the protein in its 3D structure.
Okay. And another thing that can affect the rate of the reaction for an enzyme would be pH. So here you would have an enzyme that has an optimal pH around 2. And these would be the types of enzymes in your stomach where it's very acidic, so they need to work at their best in this environment. So you can see that leading up to this pH, the protein isn't fully it's partially denatured. And once it reaches its optimal part, it will work at its fastest, and then coming back down, it's being denatured again because it's out of its optimal pH. So we already know that enzymes are able to lower the activation energy of a reaction. However, they are unable to make a non-spontaneous reaction spontaneous.
In order to understand how spontaneous reactions get their energy, we have to understand a process called energy-coupled reactions. So if we look at this spontaneous reaction, it's spontaneous because it's going from a higher energy to a lower energy state. This occurs through the release of a phosphate group, a high-energy molecule, that can then travel over to the non-spontaneous reaction.
So this phosphate group gives a non-spontaneous reaction more energy. So essentially, it raises the energy, and now we're going from higher energy to lower energy. An example of this would be polymerization.